it is time for us to move on to macromolecule number 2. Let's get into it. Lipids. That's our second biomolecule. Let's get into it. So lipids. What are lipids? Lipids is another word for fat. They are the molecule that contains carbon, so it is an organic molecule, it is a biomolecule. But they are the one that does not like water. They are insoluble. As you probably know by now, oil and water don't like to mix. And oil is a version of fat. Lipids are what we call hydrophobic. And again, here come your prefixes and suffixes. Hydro means water and phobic means to hate. So they hate water. And another word for hydrophobic is nonpolar. So water is polar, and if it doesn't mix with water, then we call it nonpolar. Some examples of these lipids include oil, like in the picture where oil and water are separated, or butter, fats, waxes. And yes, we're talking about like the wax that people use to remove hair from their body, or just wax from like candles. That is a lipid. I know you probably don't think about it as fat, but it is technically fat. Phospholipids, for example, so we talked about the cell wall in the last class. Phospholipids here are what's in the cell membrane. And some cell membranes are something we're going to go into more detail in a unit to come. I believe it's our next unit. So be on the lookout for phospholipids. So remember, the information on lipids is going to have to do with that cell membrane later. We also have triglycerides. That is the most common polymer that we normally talk about. It's also something that you get checked up at the doctor, especially when you start getting older like me. You have to make sure that your triglycerides are not too high, or that could lead to some illnesses that you definitely don't want, especially when both sides of your family suffer from those illnesses. Thank you so, Mom. Did I say thank you so, Mom? Thank you so much, Mom and Dad. So lipids are our macromolecule. The monomers, here it's something interesting because it's not just one thing, but it's a combination of two things that make the monomer. It's glycerol and fatty acids. So as long as there is one glycerol and one fatty acid together, that is the monomer for a lipid. That is a lipid. However, the most common one that I mentioned earlier, the triglycerides, that means there's 3 fatty acids. So that's the picture that you see right there. The triglyceride is just the fact that there's 3 lines of it, right? But all you need to make the monomer is 1 glycerol and 1 fatty acid chain. Now, these are the ones that tend to be the longest of all the 4 macromolecules. You have the glycerol and the fatty acid into a quite long chain. You see those dots in the fatty acid chain? That means there's a lot more stuff in there. It's not just that small. Now, lipids also contain carbon, hydrogen, and oxygen. These are your 3 main elements that we're working with so far. We'll start adding some more on later on. But carbohydrates and lipids are both composed of the same 3 elements. It's just that you see they are structured differently, and that's what makes the biggest difference between each of these macromolecules. They have the same basic elements, right? They have their starter Pokemons, the carbon, the hydrogen, and the oxygen. That's like Squirtle and Bulbasaur and Pikachu, right? But later on, we start adding more stuff on top. But without those 3, you can't start it. You can't have these biomolecules without carbon, hydrogen, and oxygen. Now, the way they're assembled, the way that you use them in the chains, that's going to make a difference between whether it's labeled a carbohydrate or a lipid. So remember, the carbohydrates had those hexagonal shapes, whereas these lipids look more like a chain. And they don't have that 1-to-1 ratio that the carbohydrates had. Here, an example of a glycerol and fatty acid combination could be C16H32O2. So there is no real correlation between these. However, the main thing that you'll notice with lipids is that there's a lot of hydrogens, and I mean a lot of hydrogens. This example with 32 hydrogens is pretty common, and you may see some with many, many more hydrogens. So don't be scared if you see like H50. That is perfectly normal. There's a lot of hydrogens in lipids. Now there's two main kinds of triglycerides or polymers of these fatty acid chains. There's the saturated fats and the unsaturated fats. Now, you may have heard these words before. Some of them you need, right? Some of them are not good for you because they're not natural and they are much, much more hard to break down. So let's get into those. The saturated lipids are the ones that are considered the bad ones. So they are saturated with hydrogens. That is the main idea. They have so many hydrogens everywhere. There's a lot of those H's. And they are usually solid at room temperature. So things like shortening and butter and lard. Manteca is so good, but you shouldn't eat it. And like, it's bad for you, but like you just put some on tortillas with like a little bit of salt and it's so delicious. And I should stop talking about this because it's past 9 p.m. and I don't need to eat anymore. So the main thing you can remember is that all the bonds between the carbons here are single lines. So they are one long chain, just carbons, right? Connected to each other by a single bond. They are saturated with hydrogens. Every single carbon has hydrogens around it on every side. And so the main issue here is that these solid at room temperature fats are bad for you because your body has a harder time breaking them down. Now, the unsaturated fats are what we think of as good fats, like avocados and olive oil and peanut oil, except if you're allergic like me, then that's not good for you either. So these good fats are liquid at room temperature, like olive oil, and they have a double bond in between a few of the C's. And so they have that slant. They're tilted, right? Have a degree angle between some H's and the others. That slant is what makes them not be saturated, because if you can tell, since there's a double bond between those carbons don't need two hydrogens. They only need one to complete their happy bubble of full, stable electron outer shell, if you remember from chemistry. So since they're happy with just one hydrogen, they are not saturated with hydrogens. Does that make sense? So they're not full of hydrogens everywhere just because they have extra space. They don't have extra space because they're double bonded with each other. And so there's less hydrogens than in the previous one. If you remember the previous one, there's 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11 hydrogens on top, 11 on the bottom. Now here, there's 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11 on top, but there's 1, 2, 3, 4, 5, 6, 7, 8, 9 on the bottom. So exactly the two that are missing from each of these two carbons that are double bonded. So there's less hydrogens. It's less saturated, less filled up. Saturated means filled up, right? So there's less filling of hydrogens. That is what makes them be good for you. Doesn't mean that you should go chug a bottle of olive oil. Trust me, that's not a fun time, but they are the good fats that you should consume every once in a while because your body does need some lipids. So why does your body need lipids? Let's talk about that. They are our source of long-term energy storage, and I've been long-terming storaging a lot during this quarantine. It is the fat that takes a longer time to break down. So it does work as kind of a reserve, you know. You have 1 gram of fat is equal to 9 calories, but it is harder to break it down. So it takes a longer time for your body to use it. However, 1 gram of carbohydrates is around 4 calories, so it's less than half, but your body's able to break it down a lot faster, so you're able to use it right away. So those are 2 of the main differences there. So it's a long-term energy storage. You're going to use it a while later. For animals like this walrus, where they might need to go away for a while, or they don't know when they're going to eat again, they need those kinds of macromolecules. They need a lot of lipids. They also provide insulation or blubber, right? That like keeps you warm in the winter for maintaining your body temperature. Normally you see very, very big animals that have to hibernate. They consume a lot of fats, and they're able to, 1, have a long-term energy storage of food that their body can break down over a long period of time while they're away. And they also have insulation to withstand the cold. It's also a cell membrane component, which, like I said, we'll talk about in the next unit. And it's also used as steroids. Lastly, it is to waterproof surfaces. So, like we said, water and fats don't like each other. They won't mix. So when you use a lipid on top of a surface, it is not letting the water seep through. So animals that look slippery, they have a lot of wax on their skin that is able to keep them dry. It's also in a lot of plants. The plants that you can tell that have like the water droplets on top, it's not really getting absorbed into the leaf. That's why they have a waxy coating. So waterproofing, that's also what we use lipids for. That is it. So on that note, answer the questions below. And if you have any questions, remember, questions are below only if you're looking at this through forms. If you're not, go back to the hub and click on the link so you can answer the questions. If you have any questions, I'll answer them tomorrow in class. Write them down so you don't forget them. And I'll see you tomorrow. Bye.